Hello, honorable judges. My name is Austin Lin, the team leader.
Place, I want to introduce the content of our project. The first one is a background, then the description, the project goal, the experiment, and the result analysis. After that, we will talk about the human practice and safety considerations. Eventually, we will introduce some of our future plans on how to improve our project and show you the attributions of each team member during the process of the project. Finally, we will show the reference of some materials used in our project. Second of all, it is about the team constitution of our IGM team. There are mainly two parts in our team, the research part in the laboratory and the business part about marketing. As for the research part, we constructed plasmid during the experiment in the laboratory and also tested and analyzed the final results. And as for the business part, we think about the way to package our final product and also do research in the marketing by investigating people from different walks of life and consulting those experts. And through the conversation with those experts, we got a great many useful suggestions and inspirations. The very high prevalence rate of galactosemia is really a very serious problem. About 1 out of 60,000 people nowadays in the world have the possibility to get this kind of disease. And this prevalence rate is lower in Asian countries, and this prevalence rate is higher in American and European countries.
Firstly, we have to introduce the background of our project. Galactosemia is a genetic disease resulted from abnormal galactose metabolism. This disease can cause clinical manifestations such as vomiting, refeeding, restlessness, and diarrhea in a newborn after taking breast milks, followed by decreased muscle tone, jaundice, and hepatomegaly. It can even develop into cirrhosis, hypoglycemia, cataract, physical, and intellectual development disorders. As nowadays, there are 3 mainstream therapies to treat this disease: decrease the intake quantity of products with galactose, increase the intake of relative enzymes, and treat those complications when fetus patients grow up. However, they are not proved to be highly effective and efficient. To cope with this disease in an effective and direct way, we aim to build a probiotic with the function to decompose the galactose into useful butanoic acid. Our plan is to engineer 4 different plasmids using conventional methods in molecular biology to create a genetically engineered probiotic. The first plasmid will be the one that contains a butyric acid synthesis pathway, in which gives the host bacteria the ability to break down galactose into butanoic acid. The second plasmid will be the one that contains LAC-Y gene and P15A carrier, giving the bacteria the command to prioritize the use of lactose over glucose, which then breaks down into galactose and glucose for further digestion. The fourth plasmid will be a fusion of the two, allowing one single plasmid to contain both such abilities, and is also our desired outcome. However, we also plan to make another plasmid number 3, which is similar to the first one but have a smaller P15A carrier, allowing an easier transfer into the E. coli cell.
Our goal is to successfully produce the fourth plasmid and transfer it into E. coli 1917, enabling the bacteria to survive in the patient's small intestine to break down the galactose. If the first plasmid is not successfully produced, we will use plasmid 2 and 3 together as a backup, ideally having similar abilities as the first plasmid. To test their abilities, we will take them for genetic sequencing and also ferment the bacteria with such plasmid to check if a decrease in specific carbohydrate concentration can be seen. We sincerely hope that our product can help these patients, helping them break down galactose into butyric acid and open up a whole new galaxy for food products for the patients, hence our name Galactose.
So this leads on to our methodology overview. 1 contains a butanoic acid synthesis pathway with promoter J23100 on the backbone PMTL83151. First, the plasmid without a promoter is extracted from the pre-prepared bacteria solution. This provides individual plasmids for the later add-on of the promoter. Then, a KBN1 restriction endonuclease cut is done to provide the space for the insertion of the promoter. The cut creates a cohesive end of CATGG from the 5' end to the 3' end that corresponds with the cohesive end of our promoter J23100. After the cut, a gel electrophoresis is done to the DNA chain to examine whether the cut is effective and accurate. For the insertion of the promoter, annealing is firstly used to attach the two single strands of promoter into a double strand one through the complementary bases. A T4 connection enzyme is then used to ligate the double strand promoter with a gap that was cut before on the backbone before the butanoic acid synthesis pathway. Here, we have already got a fully functional plasmid. It was then transferred into a competent E. coli DH5 alpha cell for further replication to create more samples. Colony PCR and DNA sequencing were finally done to test the accuracy of the insertion and ligation. The results were promising. This leads us to our second main checkpoint for our research, creating our second plasmid that contains the LAC-Y gene and the P15A carrier. The first thing we did is to amplify the LAC-Y gene from a commercially produced bacteria with the gene. We achieved this by culturing the bacteria using a Petri dish and add specific primers on both strands of the gene to go through PCR using Primestar enzyme. This causes the LAC-Y gene to be amplified into large amounts, making it easier to get the gene in high yield and concentration. Moreover, we did the same to the P15A carrier gene, amplifying into large amounts for easier manipulation. Then, we tested the results of our PCR by letting it go through gel electrophoresis and took pictures of the gel under UV lights to inspect the glowing bands of DNA for any abnormalities. We then took the bands with the correct gene and cut it out for DNA extraction from gel in order to finally obtain the LAC-Y gene and P15A carrier. However, the LAC-Y gene is not yet finally complete, as we have to add a promoter before the LAC-Y gene to let it express itself. We achieved this by doing a process to overlap PCR, overlapping the promoter 2J23200 forward with the LAC-Y and amplifying the resulting template. This has again gone through gel electrophoresis for the testing of the results. Then we had to fuse the LAC-Y gene along with the promoter with a P15A carrier to create a finally and fully functional plasmid number 2. This is achieved using the multi-S ligase to connect the two genes under warm temperatures. After the ligation process, we had transferred the plasmid into a competent E. coli cell, DH5 alpha, and cultured it on Petri dishes under chloramphenicol antibiotics. As the plasmid too also have genes that decompose the antibiotics, we were able to use this ability to only culture the bacteria with the plasmids. The bacteria without a plasmid will be unable to survive under the antibiotics, leaving only the bacteria with the plasmids behind. Then we picked the samples from these bacteria colonies and put them to a fermentation test. Meanwhile, the samples were also taken to a biotechnology company for gene sequencing to test if it is indeed the right plasmid. The fermentation test is carried out by preparing M9 solution mediums with the glucose, galactose, or both as the solution. This test determines whether our plasmid construction is a success and whether a plasmid can successfully complete its desired task. The results will be discussed later in our presentation. Plasmid 3 is a backup plasmid that contains plasmid 1's butanoic acid synthesis pathway plus the promoter with a simpler carrier, P15A. PCR is firstly applied to plasmid 1 and the P15A carrier. After the amplification, the samples could have enough amount for further modification. Because we didn't want the backbone PMTL83151 on plasmid 1, we used the TPN1 to cut the backbone into smaller pieces by demethylation modification. For the ligation, multi-S is used to ligate the fragment and the carrier into a closed-loop plasmid. The plasmid would then transform into a competent E. coli cell DH5 alpha for further replication. After that, colony PCR was done to check the accuracy of ligation. Finally, the plasmid was then transferred into an E. coli Nissl 1917 for a fermentation test. The final step of our research is the construction of plasmid 4 that have both the LAC-Y gene and the butanoic acid synthesis pathway. We achieved this by amplifying plasmid 1 and plasmid 2 once more to achieve a very high yield for easier manipulation. Then we used the multi-S ligase once more to ligate plasmid 1 and plasmid 2 to create the final plasmid. The product went through PCR once more to obtain a higher yield and is then tested using gel electrophoresis. Unfortunately, we weren't able to succeed in making the final plasmid on our first try.
The goal of our experiment is to break down galactose into butanoic acids. Because of limitation of time and resource, the ultimate plasmid that contains the butanoic acid synthesis pathway and the LAC-Y gene integrated in P15A carrier wasn't successfully constructed. After the fermentation test for the plasmid that individually contains butanoic acid synthesis path and LAC-Y, we use HPLC to measure the concentration of butanoic and acetyl acid in the bacterial solution. Unfortunately, no butyric acid is measured in any of our samples from 2-hour to 24-hour fermentation. The problem may be that some substance or variable affects the full expression of genes. Among the bad news, some good ones prevail, and it is that first, the plasmids that individually contains butanoic acid synthesis pathway, or LAC-Y, were constructed and were proved by the results from colony PCR. The exact results are shown in the slide. Furthermore, the results of gene sequencing also accurately in line with expectations. The exact results are also shown in the slide. And there are some patterns in the result that show that the modified plasmid has stronger ability to break down galactose than wild types. According to the graph above, the E. coli Nissl 1917 with butyric acid synthesis pathway leads by the promoter J23100 integrated into P15A carrier, and the one with LAC-Y fragment has stronger ability to break down galactose in the presence of solution with only galactose or the one with both glucose and galactose compared to the wild types. Solution with modified E. coli Nissl 1917 has galactose concentration decreased to a larger extent after certain time than the wild types. These above results are very promising as they show that our gene successfully gets their job done and is able to digest galactose that can be deadly for the body of the patients. However, the COVID-19 pandemic created significant time limitations for our research, so plasmid number 4 wasn't correctly synthesized. The butyric acid synthesis pathway, although cannot produce butyric acid, does perform the most vital job of breaking down galactose efficiently. Thus, these results show that the probiotic, if having both plasmid 2 and 3, will be able to prioritize the digestion of galactose.
To start with, I would like to tell you about the aim that we want to accomplish through the human practice, and then I will show you more details about how we conducted our human practice. One of our paramount goals of human practice is to raise the awareness of public toward galactosemia. In order to do this, in the first place, we interviewed some doctors and commercial leaders in the field of milk products and medicine and got a number of refreshing ideas and suggestions about our research. We conducted an interview with Dr. Lee, an expert in the field of galactosemia. We asked some questions about the present circumstances of this disease, such as the mainstream therapies and the prevalence rate of this disease. Most importantly, we got an inspiration to make our final product in the form of health beverage and sweet medicine so as to make it more acceptable for baby patients. After getting those very vital informations, we started to conduct our own survey. Our own survey mainly contains questions from two aspects: public recognition to galactosemia and their attitude toward our research project. According to 440 investigation papers we eventually collected, our assumption to make the final product in the form of RM medicine and house beverage are supported by the result. As was shown in the survey, over 60% of people from different walks like medical workers, students, office staffs, and education workers supported the design of RM medicine, and over 20% of people in those walks supported the idea of making house beverage. This provided us with significant confidence to follow our initial direction. First of all, in order to raise enough funds for the designs of external products, we connected with a local English education institution and got 500 yuan as a sponsorship money for online and offline advertisement. In order to make our plan more accessible and to implement our design, we wrote a business plan mainly include the overlook of national economic situation, analysis of medicine and beverage market, and a SWOT analysis to discuss the practicability of our product in depth, which shows a good potential future for our product. For more information about our business plan, you can also visit our wiki page. To advertise our project and raise people's awareness of galactosemia, we ran both the online and offline advertisement. To complete the online advertisement, we posted articles about the introduction, investigation results, and offline sales on WeChat platform, and those introduction videos on Bilibili platform. Till now, articles on our subscription have been read for over 3,000 times. And videos on other platforms have been watched for over 300 times. According to the result of our investigation, the acceptance of our study was increased in a large scale, especially for students whose agreement has surpassed 80%. While running the offline advertisement, our teammates went to Changtai Plaza to conduct a face-to-face investigation with some small incentives such as souvenirs. What's more, selling our external products with team logos on them helped to spread out the popularity of the team, and we also earned a small bunch of money through selling those external products as a fund of our project. In the process of the research, we built up collaboration with Team Neptuner, another IGM team who is intended to make their final products into the form of house beverage. We communicated about the projects and finally put forward the idea to put those two kinds of probiotics of our projects into the same beverage. In this case, this kind of beverage will have the impact on those people with a lack of sleep and also help to decompose galactose. For the safety concern, the experiments were carried in a proper molecular biology laboratory. During the experiment, all personnel wore proper lab coats, pants, closed-toe shoes, gloves, and goggles, which were all disinfected and disposed in biohazard containers. Instructions and protocols were strictly followed when using instruments like PAPTIS, electrophoresis tanks, PCR machines, and centrifuge.
In the future, to further improve our research after the pandemic, we will need to find out how to fully express the butanoic acid synthesis pathway to produce butanoic acid. Moreover, we will also need to engineer plasma-4 successfully if more time is given. Last but not least, we'll need to simulate a digestive system environment both in alkaline and acidic environments, as the probiotic needs to survive in the stomach and small intestine.